Good afternoon, and welcome everyone to the Arthritis National Research Foundation's Research Spotlight webinar series. I'm Amanda Giannino, VP of Development with ANRF, and on behalf of our Board of Directors, Scientific Advisory Board, and our staff, I would like to welcome you to the Spotlight on Lupus. We are so excited to have many of you joining us from all around the world. We would also like to recognize and thank each of our corporate partners for supporting our Research Spotlight series. Thank you, Amgen, for being our gold level supporter for this series. And thank you to AstraZeneca, AbbVie, and AOPI for your continued partnership and dedication. It is because of you and your partnerships that we are able to continue to pave the way for new innovations and learnings from our researchers. Thank you. Now, before we get started, there are a couple logistical pieces of information we would like to share. This webinar is being recorded, and we will have it available on our website and YouTube channels in the upcoming weeks. If you have questions, please type them into the Q&A. If you aren't able to get to yours, please email info@curearthritis.org after the webinar with your question. Now, I am honored to turn the mic over to our patient moderator, Alita McLean, to tell us her story living with arthritis. Alita?
Thank you, Amanda. Good afternoon. My name is Alita McLean, and I'm a person living with lupus. I was diagnosed with lupus in 2014. When I was diagnosed, I was scared, angry, and lost. Imagine losing your hair and being told it was stress. Imagine going to the doctor for flu-like symptoms, but being told you have a bladder infection. Imagine having discoloration all over your face and being told you're having an allergic reaction to the medication they gave you for the bladder infection that you end up not having. Imagine that you end up finding out your liver was inflamed. Imagine waking up in the middle of the night to turn on the air, and as you walk, your legs give out on you. Imagine going to ICU, being screened by multiple specialty physicians, but still no diagnosis. Imagine being told we will have to do a bone marrow biopsy because your white blood cell count continues to drop. You're at a high risk for infection, and we still don't know what's wrong with you. Then imagine being told you have lupus, there is no cure, and you'll be on medication for the rest of your life. I didn't know anything about lupus when I was diagnosed. After being told there was no cure and learning the side effects of the medications, I became scared and worried. I had so many questions. Will I be able to go back to work? How do I explain this to my child? But to help with some of my concerns, I joined a support group. And to hear other women who were 20 plus year survivors and some who were just as scared as I was, gave me hope. This also gave me the courage to advocate and to educate others. Telling my story helps free me from my pain, and I hope it gives someone else the courage to tell theirs. I want to thank the Arthritis National Research Foundation for allowing me to tell my story and for hosting this webinar. And without further ado, I will introduce our speakers for this afternoon.
Thank you so much for that introduction and really for sharing your story. I think Shaun and I both do research, but we also see patients. And so, your story really rings true for us. So I'll begin this by talking a little bit about lupus in general. Then I'll hand it over to Shaun for a bit, and then I'll talk a little bit about my research. So you already kind of know about me, but I grew up outside of Baltimore, Maryland, went to undergrad at the University of Maryland, and then moved to University of Pittsburgh, where I really completed the rest of my training and have continued my research career. I got into lupus and rheumatology because I enjoyed the medicine. I enjoyed the interaction with the patients, and also just because of kind of family members with autoimmune disease, which really drew me to this area and the kind of underserved population that's in it. In addition to kind of work, I also enjoy soccer and spending time with my children. So a little bit about SLE, or as we all refer to it as lupus. So this is an autoimmune disease that really can target any organ in the body, and I'll talk about that in a minute. But for each person, the disease can be quite different. So as we know, we kind of describe every patient as a butterfly because they all are a little, or a snowflake because they all can be different. Lupus can affect the skin and blood counts. Other can affect the brain and the kidneys. And all lupus patients, though, have something called autoantibodies. And so autoantibodies are signs that your immune system is really reacting against something in your own body. And so many of you, if you're lupus patients, will have been told by a doctor that you've had a positive ANA test, or maybe even a more specific autoantibody against double-stranded DNA, or one of the other things that we usually test for. And unfortunately, we still don't know the exact cause of lupus, but we know that there's numerous contributing factors. And this can include genetics, immune dysregulation, environment, but also random events. And so we're trying to get a better understanding of all of these aspects to really understand the disease. So as far as lupus symptoms, there's many different kinds of rashes that are associated with lupus. Some of our patients will have discoid or scarring rashes. Others will have diffuse rash all over their body, or hair loss, or oral ulcers. In addition to that, people can have systemic symptoms like fevers or weight loss, as a kind of our patient described. And then really organ-specific manifestations. So arthritis, so swollen joints, similar to what we see in our kind of rheumatoid arthritis patients. In addition, we have, our lupus patients can have sun sensitivity or severe fatigue. Sometimes it can cause memory and other cognitive issues. Our patients have a high risk for blood clots and stroke, and this can be associated with a syndrome that is more prevalent in lupus patients, which is called antiphospholipid syndrome. Then we have lung inflammation and low blood counts. And one of the areas that, Shaun and I both focus on is lupus nephritis and the kidney problems. And this is really the most common end organ manifestation of lupus, and associated with a lot of morbidity and mortality. And over time, we and others have worked to try to understand this better, and new therapies are coming online, which I'll talk about briefly in a minute. So who gets lupus? Well, the estimates of how many patients have lupus really varies quite widely. But there was a good study that was done about 2 years ago, which suggests probably a little over 200,000 people in the United States have systemic lupus. This does not include the additional people who have just the skin manifestations of the disease. And lupus is more frequent in certain populations, specifically in Native American and African American populations, followed by Hispanic populations, and then is a little bit less frequent in Asian and white populations. Not only is the disease more common in Native Americans and African Americans, but it also has a higher degree of severity, and lupus nephritis is more common in those populations. And lupus is far more common in females than males. It's actually 9 females to 1 male have the disease. Now, this is a little bit different in pediatrics, where it's closer to 3 to 1. But for the majority of our patients who get it diagnosed in adulthood, these are female patients. And unlike a lot of diseases that doctors treat, lupus is a disease of younger individuals, often diagnosed in childhood or early adult years. And as we kind of talked about, lupus is diagnosed by a rheumatologist, and it's based on a combination of the symptoms that we just talked about, but also laboratory testing, like the ANA, inflammatory markers, and complements, which you may have heard about your doctor talking, or heard your doctor talking about. So how do you manage the disease? Well, there's some daily things that we can do. We know that one major contributing factor is exposure to UV light, which can cause damage and seems to cause flares, particularly in the skin. Also staying healthy and staying active with exercise, and also trying to limit stress, which I know is really hard when you're suffering from a chronic disease. And then we have numerous treatment options. So as many of you are probably lupus patients would be on hydroxychloroquine, also known as Plaquenil. And nearly almost all of my lupus patients are on this medication. And we have really good data that this can help with a lot of outcomes. And then we have non-specific disease-modifying agents that we've used for the last 40 years or so, including azathioprine, methotrexate, and mycophenolate mofetil. Now, one of the nice things about being physician treating lupus patients and a researcher is that we've had kind of new agents approved in the last about 10 years. And this includes belimumab, which targets B cells, anifrolumab, which targets type 1 interferon signaling, and voclosporin, which targets T cells. Both belimumab and voclosporin have actually been approved for lupus nephritis. And so, I think as a physician who's treating the disease, this offers us a lot of hope that we have new therapies to offer our patients. So in summary about general SLE is that it presents in many ways and can affect all parts of the body. And while a major part of lupus susceptibility is genetic, environment and random events in the cells and other undiscovered factors can also play a role in the disease. Lupus is more common in women and minority populations, and there are new therapies for treatment of lupus, but there's still a lot of work we can do. So as was already mentioned, my research really focuses on three major areas: signaling in B cells, in T cells and how they change in the target organ, and then how the body tries to protect itself from disease. And just for the focus of this, I'm going to talk about a little bit about B cells. So what are B cells? So B cells are a specific type of immune cell, which is part of what we call the adaptive immune response. So it has a more specific response. And B cells do kind of three major things. So in this cartoon, you can see that the B cells are secreting antibodies. And this is what we call humoral immunity. And these antibodies usually bind to bacteria or viruses to engulf them and stop them. But in lupus patients and some other autoimmune diseases, B cells recognize our own body and create antibodies against self. And that's what we measure when we look at ANAs. And we think that these could contribute to disease. In addition, B cells secrete cytokines. And these cytokines are inflammatory mediators which signal to other cells and can damage or rev up the immune system even further. And then finally, B cells do this thing called antigen presentation. And what that does is it kind of tells the body where a bad protein is and engages this with another part of the immune system, the T cell. And together they attack those bad particles. And in lupus patients, unfortunately, those same things that are causing antibodies against self can also cause this interaction against self proteins. So we think B cells can act in numerous ways to drive inflammation and autoimmunity. And as I said, they're thought to be major contributors of disease. And one of the major things that we measure as rheumatologists are the ANAs and antibodies. And so here's a picture of different ANA patterns, which you may have seen. A lot of our patients have homogeneous or speckled patterns. And this is a general ANA test, but we can do more specific testing for other antibodies that you may have heard about, including double-stranded DNA, RNP, Smith, SSA, and SSB. And we know that these immune complexes with antibodies and proteins can end up depositing in tissues, particularly the kidney and the skin. So with that, I will hand over the presentation to Shaun Jackson.
So my name is Shaun Jackson. I'm, as mentioned before, I'm a pediatric rheumatologist and pediatric nephrologist at Seattle Children's Hospital. I originally came from South Africa and then moved and did medical school in Cape Town in South Africa, and then moved to the US for both pediatric training and then for this unusual combined fellowship. And I think that the slides you've already seen thus far go through why I chose to combine these fields. And the reason is because a disease that both Jeremy and I are interested in from a treating patient standpoint, but also from a research side, is lupus. And lupus has this propensity to cause systemic disease, but also affect the kidney. And so I was really interested in that really since medical school, and so decided to combine these fellowships. Unlike Jeremy, I'm not a talented soccer player, but this is a photograph of Cape Town, where I grew up. And right down in the bottom there, you can see the stadium that they built for the 2010 World Cup a few years ago. So what is lupus nephritis? So just the term, anything that ends in itis means inflammation, and the nephrology is the study of the kidney. So lupus nephritis is inflammation of the kidney. But what is a kidney? So I'm a pediatrician, so I have to explain this a lot to kids. This might be too basic for many people on the call, but the kidney is what produces urine, and it produces urine in order to clean the blood and excrete waste. So essentially what happens is that blood flows into the kidney, it gets filtered by the structure, then returns to the body, and all the waste goes out via the ureter, which drains into the bladder. And if we look at a high magnification image, the kidney is made up of about 1 million structures, which we call the nephron. And what the nephron is a filtering area of the kidney, which is called the glomerulus, which makes sort of the earliest form of urine, which we call urinary filtrate. And then that urinary filtrate traffics all, goes all through this tubular structures, which are all these specialized cells which essentially retain everything that the body needs and allow everything that the body doesn't need to filter out. And so we get rid of toxins, but also maintain things like your sodium levels in your blood, potassium levels in the blood, as well as things like the blood pressure. And so what happens when lupus happens in the kidney? Well, if the kidney is inflamed, there's certain symptoms that develop. So one of the things is that we get blood in the urine. Sometimes that's blood that the patient can actually see, but other times it's just recognized on a test. And the sort of difficulty in diagnosing lupus and being told that you had a urinary tract infection is actually not that unusual, because as you can imagine, for a primary care physician who is seeing patients and identifying patients with the blood in their urine, the most common cause of that is a urinary tract infection. However, it could also be a manifestation of inflammation in the kidney. Because the kidney controls fluid balance, one of the features you can get is swelling, which we call edema. And because it controls blood pressure, you can get elevated blood pressures. And we diagnose lupus nephritis in a patient with lupus who has any of the laboratory studies which show evidence of the kidney being inflamed. So the things we look at is protein in the urine, which we call proteinuria, and evidence of reduced kidney function, which we recognize by elevation in a blood test called creatinine. And how do we treat this disease? Well, the first thing we want to do is control the inflammation. We effectively want to treat the lupus, and specifically treat the lupus that's inflaming the kidney. But then also we want to prevent kidney damage, because we know that if this inflammation goes on unchecked, slowly but surely, chronic damage accumulates in the kidney, and ultimately that can lead to the patient requiring something called dialysis or a kidney transplant to treat the disease. So we've already talked about some of the immunosuppressive medications for lupus, and to make it simpler, I could say that in many ways, a lot of these therapies are the same as we use for what's known as extrarenal or outside of the kidney lupus. So we use medications like corticosteroids, prednisone. All patients should be on hydroxychloroquine, which we've already mentioned. Then we have our standard immunosuppressant medications. So MMF, which is mycophenolate mofetil, which was mentioned earlier. And sometimes we still use an older drug called cyclophosphamide in patients with severe kidney inflammation. And then there are biologic medications that we've talked about before. In particular, 2 new medications, belimumab and voclosporin, which have both been approved specifically to treat lupus nephritis, in addition to treating lupus in the rest of the body. And then we know that kidney damage can cause secondary problems which worsen kidney damage. And so one of the things we know is that elevated blood pressure, called hypertension, can cause additional damage to the kidney. So we want to control the blood pressure. And we also know that protein leakage by itself can cause chronic injury to the kidney. And so we like to control that as well, over and above treating the kidney inflammation. Unfortunately, there's a class of medications called ACE inhibitors, which do both things. They control blood pressure and control proteinuria. And then finally, there's a range of new therapies. The field of chronic kidney disease outside of just lupus nephritis has actually undergone some major advances in recent years for drugs that can protect the kidney from the ongoing effect of damage. And one of the things I've mentioned here is a class of drugs called SGLT2 inhibitors, which a lot of nephrologists are providing. And so I mention that to say that for patients who have lupus as well as lupus nephritis, there can be a benefit for being treated by both specialties, both rheumatology for the disease, but also nephrology to help treat the nephritis and also think about the secondary damage that can occur if patients have kidney inflammation. So where does the kidney inflammation happen in the kidney? So 2 slides ago, I showed an image of that, what we call the nephron. And that starts with urine being made in the glomerulus and then traffics through tubules where the urine is sort of, it is altered such that only the toxins are released. And the important part about kidney inflammation in lupus or lupus nephritis is that the inflammation actually happens in both regions of the kidney. So the first thing you get is deposition of those autoantibody immune complexes in the kidney, in the glomerulus, where the blood is filtered. And that's shown on the left here. See if I have a pointer. No, I don't. On the left here is the glomerulus, so you get inflammation in that region. And what that causes is for the kidney to have blood to leak into the urine, but also protein to leak in the urine, as well as various things like high blood pressure. However, importantly in the kidney, there's also inflammation that happens around the tubules, and we refer to that as tubular interstitial lesions. And so what we see in the tubular interstitial region of the kidney is infiltrating B cells and T cells, which both Jeremy and I have studied over the last few years to try and understand what's going on. The challenge here is because the disease is so complex, having inflammation in 2 different places in the kidney is, how do you understand what's going on? And more specifically, how do you understand what's going on in human patients? So fortunately, from a research standpoint, the way we diagnose the lupus nephritis is by doing a kidney biopsy and looking at the tissue under a microscope in histology. And so that's really standard of care, that all patients who have kidney lupus nephritis have undergone a kidney biopsy. However, the way we study that kidney is we mostly just looking at how it looks morphologically, what it looks like under the microscope. And we can see a range of cells that are in the kidney, but really they just stain as blue cells, and we can't really work out what type of cells they are and what they're actually doing in the kidney. And so from a research perspective, what I wanted to highlight is some new technology that we've recently applied to really understand at a very fine detail what is going on in the kidney of a patient with lupus. And so what we did in our study is we went to our pathology department, where actually all kidney biopsies that are taken are stored almost indefinitely in the pathology lab, so we can do additional studies if new technology arises. And the new technology that I'm going to talk about briefly is called spatial transcriptomics, and specifically single-cell spatial transcriptomics. So to understand this therapy, what we're talking about here is we're working out what genes are expressed in the kidney, and we're doing that at very high resolution up to a single cell. And then we can work out what cell types are in the kidney. And so if you look on the right here in the panel labeled C, this is what the kidney looks like under light microscopy. This is what we previously used to diagnose lupus nephritis. And a pathologist can look at this and say that there are various features that are associated with lupus nephritis. These circular structures here are the glomeruli, the parts that make urine. And this here is a large blood vessel that happened to be in the biopsy tissue. However, on the right here, each of these dots is an individual cell type in the kidney. And we've worked out what cell types they are based on the genes that are expressed in that local region. And I'm not going to tell you what every single color represents, but you can clearly see that there is these sort of reddish orange regions here, which are regions of the glomerulus, and this open region surrounded by black cells, which is that large blood vessel. And so now we can look in patients with lupus and we can say what is going on in the tissue. And so this is actually what one of our pediatric patients looks like. Here, the colors have changed a little bit, but the blue are the glomeruli. And you can see all of these immune cells in the tubular interstitial region, that being the gray cells here are the tubules. So you can see all these red infiltrating immune cells. And if we work out what types of cells they are, it turns out that they are B cells, and plasma cells, which are the cells that produce antibodies, as well as some T cells. And they all form into these different sort of focal structures, which we call immune foci. And that the fact that they're all clustering together really implies that they're doing something, that they're causing inflammation. Now, obviously, there's a lot of work that's going into this, and I'm not going to go into any detail about the genes that are expressed, but there's lots of interesting things that have come from this project. However, just as one vignette, I can show how studying the kidney in this way has actually helped inform us in terms of how we might treat patients. And so what Jeremy Tilstra mentioned before is that B cells are important in lupus. And I also work in a B cell focused lab. Both of us have decided to focus on lupus and lupus nephritis, and we both decided to work on B cells. However, about 10 years ago, it was determined that a drug called rituximab, which targets B cells and eliminates them, doesn't work in lupus. So the question is, why is that the case? And so just as an N of 1 case, this is a 13-year-old girl with a particular, that was treated at our institution with a particularly severe form of lupus nephritis. And what the reason you can tell that is she actually had 3 kidney biopsies over the course of an 18-month period. We were trying to work out what was going on. And each of those biopsies showed persistent inflammation in the kidneys. And you can mention all of the drugs that we've given her, and we've talked about some of them. So she was on hydroxychloroquine the whole period. She was on MMF for period. She got that IV form of medication, cyclophosphamide. She got lots of steroid medication, and she got 6 total doses of this medication, rituximab, which should have eliminated her B cells. And despite giving her all of that therapy, her disease actually remained active. So her double-stranded DNA levels remained high for the entire period. The star is when the levels are above the limit of detection on our assay. Her complement levels remained low for 18 months, and her blood albumin level, which is a measure of protein that has been leaked in the urine, but is not, because the kidney is leaking protein, the blood protein levels drop. You can see that those levels were persistently low over the course of 18 months. Despite that, she had no peripheral blood B cells in her blood. So we were measuring her blood B cell counts, her circulating B cell counts. And for most of the times, we couldn't detect any of them. Those are highlighted in the stars here. Occasionally, they sort of popped up a little bit, but then we gave her another dose of rituximab and her levels went down. And so the question is if her B cells were eliminated, why did she still have active lupus and active lupus nephritis if B cells are important in the disease? And I think that studying this one patient using that spatial transcriptomic method actually gives us an answer. Because what we noticed when we looked at her first biopsy is she had this cluster of B cells and plasma cells. However, when we looked at her third biopsy, she still had this cluster of B cells and plasma cells labeled in orange and blue here. And that was despite the fact that we had got rid of the circulating B cells. And so I think what this answers for us is the reason why rituximab was not working in lupus nephritis may well be because although it depletes the B cells that are circulating around the body, it's not getting rid of the B cells that are in the kidney. And there's another piece of evidence of which is quite exciting, but came out within the last year and has caused a lot of talk in the field, which is basically, is there a better way to get rid of B cells in a way that we can really eliminate all the B cells in the body, including the ones in the kidney? And that's something called CAR T cell therapy. And essentially what this is it's a therapy that was designed to treat adult and pediatric cancers. Some of the first patients to receive this therapy were children, where what you do is you take the T cells out from the patient, you then alter the T cells so that instead of targeting whatever they were supposed to target, influenza, COVID, etc., they now target a specific protein that's expressed on leukemia cells. But it just so happens that normal B cells express that protein as well. And then you infuse those cells back into the body, and those engineered T cells will then get rid of the cancer cells. But because they also target B cells, that over the years, we've always wondered, is this a therapy that might work in lupus? And there was a group in Germany that last year published this study where they treated only 5 patients with the disease, but these were their patients with severe lupus that wasn't responding to any other therapy. They gave them this therapy, and what happened was their B cell counts went to zero very rapidly, as one would have expected. But in addition, their disease activity, as manifest by this score called the SLEDAI-2K, plummeted down to sort of no disease activity at 3 months. And then they remained in remission for up to 18 months now. And they did this without any other medication. So it's really quite a unique feature in terms of treating patients. In very few cases do we just stop all therapies after giving a new drug, because we know lupus is a hard to treat disease. Now, this is a new investigational therapy. It's not something that's widely available, but a number of companies are interested in it. But I think it gives you a sense of how we can think about new ways to treat the disease, and also emphasizes that B cells might be important in this disease, even if our earlier trials with a drug that we thought were depleting B cells didn't work. And with that, I'll transfer. I think that's my last slide. So I'm going to transfer back to Dr. Tilstra for some discussion on other ways to target B cells.
Great. Thanks, Shaun. That was great. So as Shaun mentioned, right, we have new therapies and a lot of reasons that B cells likely contribute to disease. And while CAR T cells are great and promising, there's still a lot of, cost and difficulty associated with that therapy, which I don't think we have time to go into. But one of the focuses of some of the research that we've recently published is, can we target B cells in specific manner? So maybe not even get rid of all the B cells, but target specific signaling pathways only in B cells and reduce disease. And so there are these molecules in a lot of our cells, but particularly B cells, called toll-like receptors 7 and 9, and they really drive those autoantibodies and the reaction against DNA and RNA, which is what many of the autoantibodies in lupus are associated with. And we know that many of the TLRs signal through this molecule called MyD88. And so we had this hypothesis that MyD88 regulates a lot of these negative effects of B cells. And if we knocked MyD88 out in B cells only, that maybe we could reduce disease. But we do a lot of these studies in animal models. And one thing we realized is, when you delete a gene or block a gene before disease onset, it's really easy to stop disease, but it's much more difficult to make disease better once it's already started. So we used a specific model here. So what we decided to do in designing this study was take these mice, and these use a very specific method where we can give a medication, in this case called tamoxifen, and turn off that MyD88 gene at any point in the life of the mouse. So what we did here was we waited till the mice had disease, which starts as early as 6 weeks, but usually by 8 to 11 weeks. And then we started treating them with this inhibitor at 12 weeks of age. And what that allowed us to look at was, can we delete MyD88 only in B cells after disease onset? And then we looked at the outcome in our mice. And so what we saw was this interesting phenomenon, that here was our kind of 12-week time point, and we saw that these mice had developed autoantibodies. But then here's our two groups. Here's our control group, which had normal MyD88, and here's our treatment group, which had MyD88 deleted at this 12-week time point. What we saw was the control group developed more antibodies over time, while the MyD88 knockout mice didn't develop any more. They didn't get rid of the antibodies they already had, but they didn't develop any more. And you can see this on our ANA slide, how much brighter, which is indicative of the amount of autoantibodies in these mice that were untreated versus these that had MyD88 deleted. But more importantly, what we saw was that if we deleted MyD88 after disease, so these tamoxifen-free MyD88 mice, that we didn't get a lot of inflammation in the kidney, and these glomeruli that Shaun had talked so much about were looking normal. But if we looked at our control mice, we saw these enlarged glomeruli, with reduced space and all of these kind of purple inflammatory cells invading the kidney, suggesting that there was a lot of inflammation that we were able to block or even reverse after losing this MyD88 only in the B cells. And then over on the right-hand side, what you can see is the lower the score here, so that glomerulonephritis or that interstitial nephritis, which we've talked about, was much lower in these kind of treated mice, suggesting that we really had a therapy that not only could stop disease from occurring, but actually kind of reverse and treat disease. And so we recently published this in the Journal of Experimental Medicine. And so what this tells us is that maybe you don't even need to delete all of the B cells, but maybe if you can target specific signaling pathways only in B cells, that you can ameliorate disease. And I think this is important because we want to limit side effects that so many of our patients have with a lot of the medications we use. One other thing I'd just like to highlight is that toll-like receptors, so I already mentioned these, lie upstream of MyD88 and direct these antibodies against, so TLR7 is the RNA antibodies against Smith, SSA, SSB, RNP, which you may have heard about. TLR9 directs autoantibodies against DNA and chromatin. And so we did another paper where we deleted TLR9 in all the cells, but then TLR7 at the same time in just B cells. And similar to our last study, if we targeted this TLR7 and TLR9 signaling only in the B cells, we got a lot less inflammation. So sorry, this is a little bit blurry, but you see all these inflammatory infiltrates and large glomeruli, which are absent in the mice that have absent TLR7 in the B cell compartment. And again, we showed reduced disease by glomerulonephritis as well as interstitial disease. So we think we have several targets in B cells, which I think now are even by the CAR T cell study that was introduced, really show potential for possible future treatments and future therapies in lupus. So in summary, this work really highlights the importance of B cell and lupus pathogenesis. It also tells us that there are specific signaling pathways which can be targeted in the B cells, so MyD88 and toll-like receptors, and that B cells aren't necessary just to start the disease process, but really are required to keep the disease going. And as I said, these may be future therapies, therapeutic targets.
We have a couple questions that came in, and I'll start with you, because you ended with talking about new treatments. And what future trends do you foresee in the field of research related to lupus?
Okay, yeah, that's a great question. And I think that one of the trends, as Shaun was bringing up, is that this CAR T cell therapy, we know that there's kind of numerous studies going to be looking at that area just because of the promising results. But I also think that we're going to have other areas really looking at more molecular pathways, right? So a lot of the medications that we've used in the past, the cyclophosphamide and the MMF and azathioprine, really don't target cells in a very specific way. So I think as we learn about the molecular pathways, that we're going to really start targeting specific pathways. But it's also important to note, right, we have these patients that are all very different. So I also think studies in the future will really take into account the different patients and how they might be different genetically or with their kind of immune profile when we decide treatments.
Dr. Jackson, I'll pose the next question to you. Are there any common co-diagnoses with lupus?
Yeah, sorry, we've also got some questions in the chat that I was thinking of asking. So co-diagnoses with lupus, absolutely. There's a lot of, we mentioned a number of the genetics that places one at risk of lupus. And one of the things that we found is that sometimes the same genetic risk factor can increase the likelihood of several different diseases. And they tend to be several different autoimmune diseases. And so if I list them, Antiphospholipid antibody syndrome is one of those. Sometimes patients can have Sjogren's syndrome as well. We occasionally even have patients who have an overlap between lupus and rheumatoid arthritis. So I think the way to think about it is because the genetic risk factors are not specific for one autoimmune disease, that one can have several different autoimmune diseases being driven by the same variant. The only thing I would point out between that is the one thing that a lot of those diseases have in common is an important role for B cells. So we're obviously biased because both of us are B cell researchers, but I think it does sort of manifest that this is one of the reasons why we have similar, very different diseases, but they're still driven by the same mechanism or similar mechanism.
Okay, thank you. And we have another question that has come in. The CAR T therapy seems to permanently knock out B cells. Doesn't this put the patient in danger of being severely immunocompromised for the remainder of their life?
Yeah, I can go in there because I introduced the CAR T cell idea. So the one thing to state is that not everybody has permanent B cell knockout. So the idea that the question is referring to the fact that you've put T cells in that are designed to kill B cells, right? And so you could imagine that you might just be left with no B cells for the rest of your life. In reality, though, that actually depends on how the CAR T cells are made. And in the German study, what happened was the B cells came back relatively quickly within 3 months. And so really it was just a complete deep depletion, and then the B cells returned. And then, the patients were maintained in remission despite effectively no longer having that therapy working. Having said that, there are some patients that are left with no B cells for a prolonged period of time. And isn't that risky? I think that that's certainly a concern. And so I think we're thinking of lots of different ways to try and address this. Ways to eliminate the CAR T cells at a time if we don't think we need them, to turn them on and off, and various sort of more complicated strategies like that. Surprisingly, however, there are patients that got CAR T cells. I think the earliest patient who was treated for cancer is now about 10 years out. And from what I've heard, she doesn't have any B cells circulating in her body, and surprisingly is doing relatively well. And I think that's just a reflection of the fact that our immune system has a lot of different arms. There's a lot of redundancy. And so although B cells are clearly important, they're not the only, it doesn't mean that patients are at incredibly high risk of infection.
Okay. And continuing on with B cells, there's another question regarding B cells, and either of you can answer. Do you think that targeting specific subsets of B cells instead of all B cells can be important for treating lupus?
So I can take that one. I definitely think there are certain B cell subsets which can be quite important. There's a population called ABCs, also known as age-associated B cells, which recently have been shown to be a major player in a lot of autoimmune diseases. And they have specific markers which might be targeted. And as kind of Shaun showed in his data, right, we saw both B cell populations, but then these plasma cell populations. So there may be specific plasma cell populations which could be targeted as well. So I definitely think there's potential for this, and if done correctly, could reduce side effects of these therapies.
Okay. There was one more question related to CAR T cell therapy. Does it target bone marrow plasma cells? Does CAR T therapy target bone marrow plasma cells?
So I should have brought that up when we talked about the toxicity, and aren't patients very immunocompromised because we got rid of their B cells? The answer is actually no. The plasma cells that produce antibody titers are not targeted by CAR T cells. And that's because when B cells become activated to become plasma cells, they lose that CD19 marker that the CAR T cells are designed to attack. And so we actually know that, and I've been involved in a couple of studies where we've looked at patients who have got CAR T cells for other reasons, for cancer, And all of them are, pretty much all of them are still immune to measles, mumps, and rubella, all of the vaccines that they got as early in childhood. And so that's probably another reason why patients are not as severely immunosuppressed as you would expect after this therapy, is that they still have all the plasma cells that their body has made to fight infections, from the period of really when they were born till the time that they got the therapy. You're accumulating immune memory to these to various bacteria and viruses in the population.
Thank you. We will stop the Q&A portion now so we can go on with the remainder of our program, and I'll introduce our next speaker. Jamie Gray is the Associate Director of Respiratory and Immunology Advocacy and Alliance at AstraZeneca. With a 20-year tenure at AstraZeneca, Jamie's experience spans the areas of research and development, operations, medical and corporate affairs, and her passion for patients is what has driven her work throughout. AstraZeneca's commitment to patients through its collaborative partnerships with patient advocacy and professional society organizations led the company to be listed #1 in the 2022 Patient View report among companies partnering with respiratory patient groups. Jamie is proud to be making a difference in the lives of people impacted by respiratory and immunology-related conditions, including her own, as she lives with allergies and asthma. Jamie lives in Lincoln University, Pennsylvania, with her spouse and 2 children. Jamie.
Thank you, Alita, for that introduction, and to my fellow panelists. AstraZeneca is really proud of our partnership with Arthritis National Research Foundation and thankful for the opportunity to participate in today's webinar. At AstraZeneca, we are redefining systemic lupus erythematosus control with early intervention that doesn't have to come with unacceptable trade-offs. Our focus is on getting biologics to patients earlier and reducing patients' reliance on steroids. Everyone should have access to healthcare, no matter who they are or where they live, and we're working to address barriers to access and affordability, innovating to deliver our life-changing medicines in a sustainable and equitable way. As we continue to follow the science in immunology, we're seeking out the use of biologics to further target underlying disease drivers. AstraZeneca aims to evolve medicine through innovation and research to ultimately drive clinical remission in areas of high unmet need. We will continue to push the boundaries of science by targeting underlying disease drivers with new modalities and applying precision medicine approach from the start of discovering medicines. Thank you.
All right, well, I wanted to say thank you so much to everyone. I'm going to reintroduce Amanda Giannino, who's going to close us out.
Thank you so much, Laura. As we wrap up, I do want to say thank you so much to our fantastic moderator, Alita. We appreciate you being here and sharing your very personal story. Thank you again. Also, thank you to our wonderful researchers for sharing their innovative work towards understanding lupus. You can see firsthand the impact ANRF has in supporting these incredible individuals in their quest to find a cure. I would like to once again thank our partners, Amgen, AstraZeneca, AbbVie, and AOPI. Your support for this webinar is much appreciated. And lastly, we will be sending our live attendees new disease fact sheets along with a survey after this event. Your feedback does help us find direction for future webinars. Please share your comments with us so we can continue providing important and relevant content to you. Thank you to everyone who joined us for this great event and for all of your wonderful questions. If you did enjoy this webinar, we kindly ask you to consider making a donation today by visiting curearthritis.org/donate. Every contribution helps support grant funding for the brilliant researchers like the ones you saw here today. Thank you again all for coming, and please remember to follow us on social media as well so we all can stay connected. Thank you and have a great day.